Hello everyone, my name is Andrew Kim and welcome to the fourth unit of AP Bio Review Session. Fourth unit is about cell communication, cell regulation, and cell cycle, more specifically mitosis. Now, I'm very mad right now because I just recorded this lesson, but it's gone. I forgot to save it. However, it's fine. It's my second time doing this, so I'm going to be even better. So when it comes to cell communication, you really just need to know four different types of cell communication, which are juxtacrine cell communication, autocrine cell communication, endocrine cell communication, and finally paracrine cell communication. Now, juxtacrine cell communication, let's talk about that. Well, juxta, where have you heard of it? Juxtaposition, right? What is juxtaposition in English? Well, in English class, you would have learned that juxtaposition is when two items or two concepts are right next to each other side by side. Then you call that a juxtaposition. Juxta means side by side or touching or tangent. So juxtacrine would be a communication between two cells that are literally touching each other. Since they're touching, instead of, sending messengers through email or whatever, tying a letter on a pigeon's leg or something, you don't have to do all those crazy things. You can literally just do knock knock, here's your message, and literally just send the message physically, right? That's what it happens. However, cells don't have messengers, they don't have messages, right? So instead of messengers, they use chemical components. So in this case, for juxtacrine cell communication, what happens is one cell literally would just send a chemical component to the other cell that is touching the cell. And this secret door, well, those chemical components or equivalent to messengers can pass through is called plasmodesmata for plant cells, and for animal cells, these secret doors are called gap junctions. So again, juxtacrine only happens between two cells that are touching each other, and how the cell communication happens is chemical components, which is equivalent to messages, will just literally move from one cell to another through this secret door. Now, autocrine. Now, when we were talking about autobiography, it's like self-made, right? Auto means self. So autocrine is a self-communication, right? The cell is talking to itself. I know it's kind of sketchy when people talk to themselves, but for cells, it's completely normal. They would release chemical components and they would receive the chemical component that they released through the receptor sites. So they send out messages and they collect the messages that they sent out. Why is this necessary? Well, even the cells need to alert and notify themselves, right? Oh, you got to split. You got to go through this phase. You got to do this. You got to do that. They have to notify themselves, right? And the form of notification, self-notification, happens through this autocrine cell communication. Third type of cell communication is called endocrine. And how endocrine works is basically endocrine communication happens between very far two cells. And because they're very far, you can't just send the messenger directly or through chemical components. What they do is they do release chemical components, but these chemical components, instead of directly being sent to another cell, it will go through a bloodstream, flow through the bloodstream, and arrive at the target cell. Now, since it flows through the bloodstream, the target cell and the releasing the messaging, the cell that's sending out the messages could be spread out farther away in our body. For example, between pancreas and liver, endocrine communication can happen. So once these chemical components arrive at the target cell, then the target cell would receive these chemical components and react based on that. Now, finally, paracrine. Paracrine happens between two cells that are close enough but not touching each other. So in this case, chemical components are released by one cell and the other cell would receive the chemical components. Instead of chemical components having to flow through the bloodstream, like endocrine, and paracrine cell communication, literally the chemical components released by one cell could be received by another directly. So these chemical components would be received by one cell, and the cell would react based on that chemical component, and thus the cell communication happens. Now, let me go deeper into this whole chemical component part, because I've been explaining how chemical components are sent and chemical components are received by the receiving cell, but how does that make communication? Well, as I mentioned, these chemical component is equal to a messenger. It's like a mail. So when a cell receives that mail, then they react based on what's written in the mail or what the chemical component is coding, right? So now, if we were to explain this from a bio perspective, here's a receptor site, and here is the chemical component, and we call those chemical components as ligand. Ligands are the chemical components that are sent and received and make the cells be able to respond based on the things coded in that chemical component. So what's going to happen is receptor site will take this ligand, and based on this ligand, some chemical reaction will happen inside the cell. For example, GDP will be converted to GTP, and this conversion will promote ATP to be converted to ADP, or sorry, CAMP, CAMP, and the CAMP will, it's like a cascade, right? It's like a chain of reaction. Chain of reaction will happen, and this chain is called phosphorylation cascade. Phosphorylation cascade is basically a cascade of reaction that happens because of this ligand being combined to the receptor site. Okay, this ligand combining to the receptor site will instigate this whole cascade of reaction, and ultimately the end result of this reaction is an enzyme being turned on and actually activated, doing its proper work. Okay, so this is how cell communication happens, and this whole process can be divided into 3 steps. First step's reception, right? When the reception site actually receives information from ligand. Second part, phosphorylation cascade, or in other words, translation. Okay, so in translation, what happens? Transduction. In transduction, what happens is a lot of chemical reactions happen because of the ligand being combined to the reception site. Okay, so transduction is this phosphorylation, this cascade of reactions that happen because of the chemical components that is being binded to the reception site. Last is over here, which is response. Of course, as a result of this cascade of reaction, a response will happen, and the response is normally an enzyme being turned on, and thus the cell serving a certain role that it used to not. Okay, so that is the 3 steps of cell communication. Okay, now that's pretty much it. And now we will move to a very specific example, which is insulin. Okay, so insulin, in fact, is an example for endocrine reaction or cell communication. Now, what happens is we have 2 organelles that you have to look at. First is pancreas, and second is liver. Now, pancreas's job is to detect glucose level. When it detects high glucose level, it sends insulin to liver. So insulin is a ligand in this case, right? It's this chemical component, it's this messenger that pancreas sends to liver. But again, pancreas and liver are far away. It's not like 1 micrometer away, right? It's decently spread it out in our body. So it can't move through any of the other 3 cell communication pathways, but it has to move through endocrine pathway. In other words, it flows, insulin would flow through our bloodstream. And once this insulin arrives at the liver will receive insulin and react based on that. How will it react? Well, if liver receives an insulin, it means that pancreas has detected high glucose level, right? So we want to decrease glucose level. So what liver does is liver converts glucose to glycogen. So now we will have less glucose in our body, right? So this is how pancreas and liver communicates through insulin, through endocrine pathway to maintain homeostasis, to maintain stable level of glucose in our body, okay? So that was one example. Now what we have to look at is we have to look at different types of regulations. So regulation. Now, there are 4 big types of regulations we have to look at. First is positive. second is negative, third is double negative, and last is feedback. These are the four big ways how cells communicate with each other. Now, positive is as one increase, the other one also increase. Okay? So for example, as I send more, as I put more time studying biology, my biology grade will increase, right? So that's a positive relation and positive cell regulation. Now, you might ask me, well, Andrew, I get your analogy, but that's not biology phenomenon, right? In biology world, where would this positive reaction happen? Well, positive reaction happens when, for example, one organelle might send a message to another organelle. When, for example, glucose level increase, it sends a message, then glycogen level in liver would increase, right? So as one component as one factor increases, if another factor also increases, that's a positive relation. Negative relation is the opposite. When one increase, the other one would decrease. Okay, so for example, the biggest example was here, right? Oh, sorry. No, just for example, this can also be seen in ecology. For example, if you have more sharks, then that would cause decrease in the number of fish, right? So when one increase, the other one decrease, that is called negative relation. Now, double negative is one causes negative, but this negative causes another negative, ending up as positive. Let's say I'm Andrew, right? I hate someone named James, but James hates Justin. Then I would love Justin, right? Because I hate James and James hates Justin. So Justin is sort of my friend because we both hate James, right? So this is double negative. Negative times negative becomes positive, right? So you see how this thing works. So for example, if we have more sharks, we have less fish. But if we have less fish, we'll have more planktons. As a result, if we have more sharks, we will have less fish, so we will have more planktons. It's a double negative because as shark increased, fish decreased, and as fish decreased, plankton increased. So both of these relations were negative relations, but as a result, as we combine two negative relationships, we get one positive relationship. As shark increased, planktons also increase, right? This relationship is called double negative relationship. Last is feedback. And when it comes to feedback, it's a bit more complicated. And we have two different types of feedbacks. First is positive, second is negative. Now, positive feedback would be something like this. I fight with my brother, okay? So this is me. This is my brother. I shout at my brother. He gets mad. He shouts back even louder. I get even more mad. So I yells at him even louder. And he gets even more mad, so he would yell at me even louder. And this continues, right? As one factor instigates the another factor, and the other factor instigates the original factor, the two factors will be constantly, continuously instigating each other, bringing the result to maximum. It will just increase, and that will just keep going on, right? That is positive feedback loop. Now, negative feedback loop is when me and my mom fights. Now, I yell at my mom. I would never do that, sorry. My mom yells at me and I get sad. I get depressed. So I become quiet. The fight ends. You see a difference here? When one factor instigates the other, but that, sorry, when one factor de-instigates, what's the opposite of instigate? When one factor inhibits the other factor, that is negative feedback loop, because as one factor increases, the other factor will decrease, which will lead the original factor to also decrease and meet at a stable point. So this is more like you'll reach a certain stable point. It will converge instead of diverge because each factor regulates themselves. Do we understand this? That was negative feedback loop. Now, that is pretty much it with cell communication and cell regulation. And now we will move to cell cycle, right? So, I mean, we all have cycles. I have a cycle. I was a toddler once, and then I grew up to be a kid. Now I'm a young adult, a teenager, young adult. I'll be a dad, and then I'll be old, and then I would die, right? Like we all have cycles. Cells also have cycles. So now today I want to talk about cells cycle, okay? Cell cycle. Now, before talking about cell cycle, we have to learn some important terms. First, chromosome. Second, chromatid. Third, centromere. Third, centriole. Third, spindle fiber. Now, what is chromosome? You really got to know what chromosome is, right? Chromosome is basically, it's where DNA is stored. It's where genetic informations are stored, okay? Then what is a chromatid? Well, chromatid, the difference between chromatid and chromosome is very small, but I would say chromatid is this one strand of chromosome, okay? So for example, if you have a chromosome like this, then you have two strands, right? But we still call that one chromosome. But how many? But there are two chromatids, okay? And what connects the two chromatid is called the centromere, okay? So that's what we have to know over here. Chromosome, chromatid, and centromere, okay? Centromere connects two chromatids, two sister chromatids specifically. We call these two sister chromatids. Why? Because they're like sisters, right? They're similar. They're just like they're like twins. So sister chromatids and two sister chromatids are connected by a centromere to form one chromosome. Now, chromosome does not necessarily have to be in this shape. Chromosome could be in this shape as well, and I will explain how they change from one shape to another, okay? Now, centriole and spindle fiber will appear later, and when it appears, I'll explain, so don't worry about it. Now, let's talk about the actual cell cycle. Cell cycle is composed of two big parts, interphase and mitosis. Now, interphase has three small parts, which is G1 phase, S phase, and G2 phase. G1 phase is cell growth. Cell is still growing. Nothing special is happening, but just the cell is growing. S phase is when sister chromatids are made. I'll explain this deeper later, so don't worry about it. And G2 again is cell growth, and more specifically, preparation to split. What do you mean by split? Well, I'll explain later, because mitosis is literally about splitting cell into two. So now, as I draw this image to explain my life, human's life cycle, cell's life cycle is basically you're a baby. Okay, you're a baby cell. Now, your cell has DNA stored in something called chromosome. Initially, your chromosome looks like this. It's single-stranded, right? But one day, it changes to look like this. Now it's double-stranded with two chromatids, right? And finally, one day, it splits. Since you have two strands, each strand will split apart, and you will have two of these nucleuses made. And ultimately, the cell will split into two. And as you can see, this looks identical as this, right? So each of the two splitted cells will go through the exact same cycle. Okay, so this is the cycle of a cell. You initially has these DNA, genetic information stored in singular strand chromosome, but one day puberty hits, you get another strand of genetic information stored. Why? Because you have to split into two so that you can split into two different cells. Now, you might ask me, why do you have to split into two cells? Well, cells need to grow. Your body has grown, right? Compared to when you were 3, you're bigger now. How are you bigger? Because more cells are made. Then how can cells be made? By one cell splitting into two. Do you get it? So you're a baby, you hit puberty, and then you split so that you can grow. Now I'll explain each step more specifically. Now, G1 is cell growth. This is when you have singular strand chromosomes. Now you're still growing because before puberty hits, you want to, like when you're like only like say 90 centimeters tall and puberty hits, that's gonna be weird, right? You gotta reach to a certain level of growth before puberty hits, right? So you're still growing at G1 phase. At S phase, this is when puberty hits and this singular strand chromosome, singular chromatid chromosome will change to two chromatid chromosome connected by a centromere again, like this. Now G2 phase is again cell growth phase. Nothing special happened. The cell just grows. Why are the cells growing? The cells are growing because before the cells split into two, right? Because I explained how the cell splits into two. Before I can be an actual adult and just go live in this brutal world all alone, I want to grow a little bit, right? Even after puberty, I want to grow. I want to learn more knowledges, more skills, more, life tips, right? So it's consider this as like a college period, okay? Like I'm just still growing. Although puberty has hit, I'm growing to prepare for my very important part of my life, which is splitting. Okay. Now, mitosis is where the actual splitting happens, and mitosis can be more specifically, mitosis includes 4 smaller steps, and those are PMAD, prophase, metaphase, anaphase, and telophase. Now what happens is in prophase, this is where our 4th term, centriole, appears. Centriole will appear, and you will have your chromosomes like this, right? So in prophase, nothing big happens. Centriole appears, and spindle fibers, these small lines, will start coming out from the centriole, okay? So centriole is where the spindle fiber comes out from, okay? And once the spindle fiber comes out, let me just charge the laptop. The cell will enter the metaphase, where the spindle fibers will actually attach to the chromosomes, like this. That is metaphase, okay? The spindle fibers actually attach to the chromosomes, and the chromosomes will align in the middle of the cell. And anaphase is when these chromosomes, these sister chromatids, will split into two. The spindle fibers will pull away, rip off each chromosome so that one chromatid, each sister chromatid, will be pulled towards the centriole. And finally, in telophase, the spindle fibers, the centriole, all of them will be gone, and the splitted chromosome will be pocketed in their own membrane. So this is how the whole splitting thing happens. And lastly, after mitosis, there's last step, which is called cytokinesis. And here, as you can see from telophase, the two pockets of chromosomes are still in one cell, right? In cytokinesis, the two cells will actually split apart into two. So that is how whole cell cycle will look like. Okay? Now, there is a reason why this cell cycle is grouped with cell regulation. It's because in the cell cycle, there are very important cell regulations happening. Now, there will be 3 big regulations, one in G1 phase, one in G2 phase, and last in metaphase. In G1 phase, there is a checkpoint. Before moving on to S phase, where the chromosomes hit puberty, there's a check phase, there's a regulation happening so that bad cells with disabled chromosomes, do not duplicate into sister chromatids because you don't want disabled cells to be replicating itself, because more disabled cells in our body means our body will function worse. So there's one checkpoint that checks before making sister chromatid whether each chromosome is healthy and worthy of making sister chromatid. The next checkpoint in G2 regulates so that before splitting, we're checking if this cell is worth splitting, right? Again, we don't want to split random disabled cells. So we check if these sister chromatids, these chromosomes, after being duplicated, is healthy and good and worthy of being split into two. Lastly, in metaphase, we check if the spindle fibers are all connected properly to the chromosomes, because if they're not, we shouldn't move on to anaphase and rip apart, right? We gotta rip off when all the chromosomes are aligned properly and it's healthy and it's ready to go. So we have 3 checkpoints, and these checkpoints are where regulation happens. So that's pretty much it for this unit. Thank you so much for watching this video, and most importantly, don't forget to have a great day.